Today we are going to talk about protein-mediated transport. Now there are two types of protein-mediated transport. There's some that do only kinetic energy, and some that need more than kinetic energy. So that probably will consist of ATP or GTP. But we're first going to start with those that only need kinetic energy, so are passive forms of transport, and that includes simple diffusion and facilitative diffusion for short. Now, simple diffusion is very simple. Well, I should probably focus on passive. So passive diffusion in general is based off of a concentration gradient. So high to low. That's the movement of particles or ions. It's from a high concentration to a low concentration. So looking at simple diffusion, we know that it doesn't require energy, so I just look at it as more stuff outside results in more stuff inside, because it's that movement. So that's the result. This is the result. Now, I like to use a graph to show the rate of simple diffusion. And because it's based off of channel proteins, are specific, meaning that some will only let sodium in, or potassium, or potentially only glucose. Now, these pores are always open. Okay, and so it's just always based off of that concentration gradient, like we said previously. Now, if we look here at the rate, it's going to be constant. Okay, it's constant. The rate is constant with time. Okay, now if we move over to facilitated diffusion, I'm going to erase some of this stuff. Okay, facilitative diffusion is determined by the concentration of the proteins. An easy way to describe facilitative diffusion, just think about as if we had a certain number of proteins, okay, and a certain amount of ions, okay. Now, the amount of ions the rate will continue to increase for a time, but then all of the protein channels will become saturated and it will plateau as such, because there's only a limited number of proteins. Some examples of these of facilitated diffusion and types of channels and carriers associated with that, we have a mechanical gated, a voltage gated, horrible handwriting, and chemical gated. An example of mechanical gated ion channels is potentially due to pressure. So if the cell senses pressure, let's say, and this would cause that channel carrier to open, right? And so allowing ions or whatever to travel through or pass through into the intracellular space. Now, voltage gated is it's based off of voltage. So if there's an increase in voltage, then that channel will open, right? Opens. Now for chemical, these gates need a specific ligand, and if that ligand binds, then it allows it to open. An example of this is acetylcholine. Acetylcholine, which is a very common ligand for chemical gated ion channels. Now, I'm going to erase this and we're going to move on to active transport. Active transport. I'm just going to delete all this because it's kind of in the, well, I'll delete that. I can't even. Okay, so there are two forms of active transport. We're going to actually describe active transport. And this is the need for energy. So, as I said previously, there are two types of energy that allow for molecules to move across membrane. With these proteins is ATP, let's do this. ATP and GTP. Okay, primary transport. It's very, very, very important. Okay, an example of primary transport is the sodium-potassium pump. Okay, this requires ATP or energy to be used in order to pump 3 Na or sodium out of the cell and 2 potassium in. Okay, with this active transport, it allows us to have secondary active transport. So bad at writing. Okay, and this indirectly uses ATP because secondary active transport depends on primary active transport to create a concentration gradient opposite of what it normally would. So I guess for some examples that we could, some two kind of subcategories of this that we could look at are symport and antiport. So I'm just going to do that down here. So symport, as in the name, it says that they will travel together. So we always got to remember secondary. One molecule travels against. Oh, I didn't write that. Against, and one molecule travels with the gradient. So, for example, if we're looking at a cell, we have sodium and potassium. Okay, now we see here that potassium wants to leave. Okay, because it's symport. So sodium goes against the gradient. Okay, now for antiport. Okay, antiport. I'll just do a simple drawing here. You got a protein. So bad. Okay. Now, one of the molecules is going to go with the concentration gradient, okay, with gradient, and another is going to go against the gradient, and that is just an example of antiport. Okay, now we already hit specificity because some will only allow specific ions or specific molecules to pass through. We hit rates. The rates will slow down. Rates will slow down as concentration decreases. That's very important to remember. And also remember our graph with facilitated diffusion, right? The rate of diffusion is dependent on the number or the concentration of proteins. Okay, now we know that active uses energy, yes. Passive does not. Okay, no. Okay, competition saturation. We already passed over direction because that was our two examples of symport and antiport. Okay, and with passive as well, that it goes from a high to a low concentration. Now, if we're studying or we're looking at competition, okay, there are some protein transports out there where a variety of ions and molecules will compete for that space. So let's see if we can draw a picture. So we have sodium. This is just theoretical. Okay, potassium. Let's just, I don't know, not water. That doesn't work. Or like glucose. Let's say they're all fighting. Okay, they're all fighting for the same spot. So it's going to be very difficult for a certain amount of, say, sodium to pass through because it's fighting against these two guys right here. Now, if glucose had a higher concentration or was more saturated, right, it would be able to pass through and outcompete the sodium and the potassium. Okay, that's very important to remember. Now, we're going to get into osmolarity and tonicity. Okay, so the main difference between osmolarity and tonicity is the fact that osmolarity does not predict how water, H2O, will cross the membrane. Okay, osmolarity is just a comparison. This is how I like to say it. Comparison between two solutions, really. And tonicity actually allows us to see what happens. So it tells us if the cell shrinks, swells, or stays the same. Okay, it's very helpful. Now, the difference between molarity and osmolarity is that molarity, of course, has moles. Let's get a different color. Is related to moles, and osmolarity is actually related to particles per liter in our solution. Okay, molarity is more related to the amount of solute, and osmolarity is actually related. Osmolarity. It's actually related to the number of particles per liter, so it's very different. Now, if we are wanting to understand how this will all play out, we have an example. So I have a 300 milli osmol solution of urea and a 300 milli osmol cell. Okay, and I guess first to understand osmosis, I can draw a picture before we get back to that. Just like in the transport, if I have a highly concentrated cell and a low concentration of solutes outside of the cell, water will move from the low concentration, so low concentration of solute, to high concentration of solute to equal out, right? So the cell would actually increase, so it would expand in water to have the same amount of concentration outside and inside. An example I would like to share is to describe how can a solution be isoosmotic. So isoosmotic, okay, yet be hypotonic to the cell. So right over here we have a solution of urea, 300 milliosmoles, right? And a cell that's also 300 milliosmoles. So that means they are isomotic. They are the same. Now, hypoosmotic, just to help you, so hypo means less, okay, root word for less, and hyper means more, okay? Now, if we look at what happens to the cell when we place it into the solution of urea, okay, you have 4 particles of urea. In order to equal out, so that's the same amount of urea on the inside and the outside, 2 things of urea will actually go inside. Okay, with that being said, if 2 particles of urea go inside, that means that there'll be 150 osmol outside, and then 300 with the cell, 150 osmoles with the urea. So, that being said, the total on the inside of the cell will actually be 450 and 150 on the outside. Okay, now this is basically concentration. Okay, if there's 150 milli osmol outside and milli osmol inside, that means water is going to move towards the more concentrated portion of that of this diagram. So water is actually going to enter the cell, swelling it and making it expand. Okay, and that is a quick rundown of osmolarity, difference between molarity and osmolarity, difference between osmolarity and tonicity, a quick brief overview of proteins and the different types and what energy is needed and how they move across